It's been great to see the presentations so far. Lovely to see all the research happening at USC and be part of the showcase today. My name is Karla van Huyssteen and my research has been focused on molecular diagnostic assays for malaria. So malaria is a life-threatening febrile disease that is responsible for almost 500,000 deaths globally each year, and it's endemic to poor tropical, subtropical areas of the world, including sub-Saharan Africa, Latin America, and southern Eastern Asia. Plasmodium falciparum is widely recognized as being the most deadly plasmodium, and it's responsible for 99.7% of the cases in the WHO Africa region. And the incidence rate of falciparum is measured as the number of clinical cases out of every 1,000 cases each year. So high incidence here is shown in yellow, whereas lower incident is represented in purple and grey, going towards white where there are 0 cases. Vulnerable populations include children under the age of 5, pregnant women and travellers or migrants because they lack immunity or have an undeveloped or suppressed immune system. Globally, efforts are taking place towards elimination. However, this is a challenging battle. 4 countries that have recently in the last, yes?
Karla, sorry to interrupt. I'm just letting you know that we're looking at your notes page, not your actual presentation.
Okay, oh dear. Let me see if I can fix that. I'll try to
switch the presenter view to your other screen.
I will do that.
If you make the presentation again, Karla.
Yep.
Just go up to the presentation mode.
Yeah.
As if you were presenting.
Yes.
Yeah.
Now what?
That actually looks good now.
Okay.
Are you not able to see your notes down?
No, I'm not able to see my notes.
That's okay. You can put it back the other way so you can read your notes. That's cool.
1 moment, sorry. Wonder how other people managed it.
I think everyone's got 2 screens going.
Oh, okay, that must have been how. Okay. Yeah, there doesn't seem to be a way around it. I'm sorry.
That's okay.
It's okay, just keep going. And yeah, that's good.
Sorry, you'll have to deal with my notes.
Thanks, Karla.
No worries. So a critical challenge to elimination is the asymptomatic reservoir. And so asymptomatic carriers are parasitemia positive individuals who lack fever or other clinical signs of malaria infection. So we can't see the classical symptoms there. And this can represent either an expanding biomass that will eventually become symptomatic disease or a chronic low-level infection that will just remain asymptomatic. Either way, this represents 20 to 50% of episodes of human to mosquito transmission. And this is a huge issue for elimination because 75% or more of people identified in community screening are asymptomatic. And these are still contributing towards perpetuating transmission of malaria. Current methods for diagnostics include microscopy, which is morphology based. It's the gold standard, and it uses stained blood smears for the detection. It requires a light microscope and a trained microscopist, and even between 2 expert microscopists measuring the same blood smear, there can be levels of error and operator differences. Also common are rapid diagnostic tests, and they detect antigen on lateral flow strips. However, these can suffer from false positives. Both microscopy and RDTs are widely applied, but their sensitivity suffers and is not good enough to detect asymptomatic levels. So newer molecular diagnostics detecting nucleic acids like PCR have lower limits of detection, which is great. However, even better than the gold standard of microscopy. However, PCR assays are restricted to central research laboratories because they involve an expensive equipment such as the thermocycler. They require electricity, they take a long amount of time, and they need skilled labor. Emerging isothermal amplification techniques like LAMP and RPA employ different enzymes to also detect nucleic acids at a single temperature, and these have a high sensitivity similar to PCR and have the simplicity of the RDT tests because they are also a point of care type test. And this raises the question, can a field amenable diagnostic be applied that is sensitive enough to detect asymptomatic infection? My research has focused on progressing a Plasmodium falciparum molecular diagnostic assay, which includes a single-step DNA extraction, isothermal amplification using RPA, and a lateral flow detection. So parasite DNA is extracted by using finger prick blood, and then we add that to a proprietary extraction buffer previously discussed by Joanne. And this causes the lysis of the red blood cells, which actually releases the Plasmodium DNA within 5 minutes at room temperature. When added to primers, probes, and rehydration buffer, the 18S rRNA gene target is amplified to detectable levels isothermally at 39 degrees for 10 minutes. And then the amplicon is then detected visually on a lateral flow strip, similar to a pregnancy test, after 5 minutes. To see if we could detect asymptomatic levels of infection, we initially tested the assay just with a plasmid to develop a baseline measure. And then we tested the parasite-infected blood, where we tested red blood cells, human sera, an actual live parasite of P. falciparum. And looking at the results for plasmid sensitivity, we see reacted strips here, lateral flow strips with a control band, and that indicates that the liquid wicked all the way up the strip, representing a valid result, and the test band here, which represents the presence of P. falciparum DNA at the concentration level that's decreasing here, our copy number. To verify the visual readout, we used electronic imaging software and an analysis to measure and standardize the black pixels of those test bands. And then we adjusted for shadows and background white space. So here, an above 0 value actually represents a result that is positive because it is greater than 3 standard deviations above the negative control average, which gives us confirmation that our positives are positive. Now, if we look at the important result, the lowest limit of detection we found here was 13 copies per microliter, and that was seen visually in 4 out of 9 results and confirmed electronically using a reader in all of our trials. Onto live parasite testing using infected blood. This is where we actually tested to see if our single-step extraction method worked. We were able to successfully detect 50 parasites per microliter, which is great news. For initial confirmation that our assay could detect all of the strains of falciparum, we tested 7 geographically distinct strains, which were provided to us by the Australian Defence Force, all of which were successful. And then after researching the characteristics of these strains, I found that 2 of the strains have a PFHRP2 gene deletion, which means that they lack expression of histidine-rich 2 protein. And that's the basis for 80% of all Plasmodium falciparum RDTs, meaning that these 2 strains would be potentially undetectable by RDTs, while our assay was able to detect both successfully. So how does our result compare and what can we use it for? So without optimization, our limited detection of 50 parasites per microliter is already able to detect less than the levels that are defined as subclinical and low density parasite infections, which actually means that we're able to detect asymptomatic parasite levels. And these levels would potentially be missed by routine microscopy and RDTs. And this shows key progress to resolving the asymptomatic reservoir that we know can sustain transmission. And then what can we use this result for? Our sensitivity also qualifies the assay to be used for case management in elimination settings, and it also approaches the threshold for being used for screening and surveillance at the district level or below. And these targets have been identified by the WHO and the Malaria Diagnostic groups. So some acknowledgements. My contributors, Associate Professor Joanne McDonald, who was my honours supervisor, Dr. Nina Pollock, USC, and as well as collaborators from the QIMR Institute, Dr. Leon Hugo, and the Australian Defence Force for our samples. Funding was provided by the NFMRI grant, the Bill and Melinda Gates Foundation, and USC. Thank you. Questions?
Thank you very much, Karla. You're certainly working on a very important issue because of malaria obviously being a real problem. Anybody has any questions for Karla? No? I think people are starting to run out of steam. All right, thank you very much. And we'll move on. Vu, can you please start your presentation, please?
Yes, let's share my screen. Can you hear me?
Yes, we can.
Yeah, that's
but
right.
We can't see your presentation.
What is now?
And don't forget to press share.
Starting to share it now.
Yes.
Yes.
But put it in presentation format, please. If you, yes, perfect.
Yeah. So good afternoon, everyone. Today I'm very happy that I would like to share with you my recent research outcome about the population genetic of striped catfish. It is belong one of my chapter. Yeah, and it's very happy to tell you that this was accepted recently last week. Yeah. So first of all, I would like to thank the team in Vietnam that research aquaculture number 1, number 2, and also the Institute Research for Genome Research for let me to access the data and help me in writing this paper. So I think many of those of you who don't know that what a dry catfish look like, so I put 2 pictures here. The picture A, the young catfish is about 3 months on, and the picture B is the commercial scale of the catfish when they get when they fillet and they make a product for international trade. So in Vietnam, the dry catfish is normally farmed in pond, earthen pond, and it takes about 6 to 8 months to reach that size of 1 kilo. In a scientific context, that the dry catfish has different names they use by different group of research, like dry catfish. But we strongly that in Vietnam we call it bangasat, and in Vietnamese we call tra, and also in Japanese they call it sushi catfish. But the scientific name is bangasat nodong and hippopotamus. So Vietnam is the first nation, the pioneer nation that put dry catfish farming for commercial scale since 2000, and up to now, and you see on the bar graph on the right that the aquaculture of striped catfish still increasing. Let's see, let's say for example that the figure aquaculture figure in 2 years ago in 2018. So we farm around over 5,000 hectares with a 1.3 metric ton. That's accounting for over 50% of worldwide production now. And the value around 1% of Vietnam GDP is cost around 2.4 billion US dollar. And we make it to export to around 150 countries and territories. And on 95% aquaculture that's go for exportation, so only 5% for domestic consumption. For the distribution in this fish is indigenous fish to Mekong River system that you see on on the map that in green area, that's the lower Mekong region. Before the trial catfish is delayed and they distributed to upper to China and Myanmar, but now due to heavy hydropower dam constructed there, so hardly really found catfish there, the upper river. And catfish in the downstream of Mekong River are now used for aquaculture and the fish trail to other countries for aquaculture purpose. So based on the literature, some research earlier, they found that one big population in the lower part of Mekong River, especially between Vietnam and Cambodia. Also, the small population at the middle part of the Mekong River between Laos, Thailand, but now it's not easy to find the fish there. And also, Chao Phraya River belong to Thailand region. They have also the more population, but the population is how to say that the proof for maybe genetic bottleneck, so the population didn't yield for aquaculture purpose. This fish have been domesticated for long, earlier in Thailand in 1967, and maybe later, 2 decades later in Vietnam. But Vietnam will make a successful aquaculture for this fish. And also the fish from Thailand, they imported to other countries like China, Bangladesh, and that take around 40% of aquaculture nowadays. So based on the distribution of fish and aquaculture in the region, we at the moment we don't know which the relatedness between the population, for example, in the lower Mekong River, especially between Laos, between Thailand, Cambodia, and Vietnam. So we here in this study we try to understand or try to answer 2 questions. So first is that about the genetic diversity of the population around the region, and based on the population genomic parameter like rapid allele on the other meter like heterozygous or homozygous allele. And question number 3 is we try to answer that how many population in the lower part of Mekong River that below Thailand, Cambodia, and Vietnam, because this region is crowded aquaculture of this species, and also the mainly habitat of fish indigenous for this fish. And number 3 that we try to answer that which population that need for the genetically selective breeding program. We try to analyze and understand other population genetic parameter like effective population size in breeding, or also the migration rate between the population to answer the question. So for the sampling information in the graph that we may see on the star, that the sample cluster we based on the Mekong River. We collect the sample, and nearby the farming location of farming pond. There total around 24 collection like this, but we cannot put a lot of style here. So the region style belong in between Laos and Cambodia River. They share the same Mekong River. And for the yellow style within Cambodia and green style in Vietnam. So we can see here, so the sampling there around 4 or 5 years ago. And we also collect the sample from Bangladesh that we believe that the fish are imported from Thailand before from Mekong River. So let's see which the disease of the genetic reduce after a long time use in there. So for analyze the data, we use the technique called the dark sequencing. That means the diversity array technology or something that look like RAS sequencing. And we acquire around maybe 17,000 numeric SNP with 93 individuals. So after high quality control, like filter for the corn rate, how they went back and discard outlier, for example, with around maybe 10 filtration like this, we acquire around 7,000 code this SNP with 90 individual this data that we use for analyze the later result, which I will show later. So we use mainly our pockets, dark and diversity, and some software, pre-software to infer the population structure and the estimator for estimate the effective genetic, effective, how to say, effectiveness, the population size, and a migration between population like this and structure. So this is the first result I want to show you, that some basic genomic parameter of its population. So here is some acronym of the population, like farm from Bangladesh, farm from Cambodia, farm from Thailand, farm from Vietnam, why Cambodia, why Thailand, why Vietnam. So we can see here the except why Thailand, they have maybe so high magnitude of the value here compared with the other population. So the main population is still close together. And also we found that the parameter from farm Vietnam and white Vietnam is nearly identical, nearly the same. So the ingredient of each population is not quite high, but the value from white Thailand is minus. Maybe they show that the white Thailand is still far away from other population, and they have high genetic diversity compared to the other. So likewise in this table, they use the FST value, the genetic diversity, total genetic diversity, to create that table. So we saw here again, the white Thailand, they stand far from the other population. And also we saw the white Thailand from farm Thailand, white Thailand, they also far. That's interesting, and we'll talk about it later. And we can see that the genetic distance between Cambodia and Vietnam here is Cambodia and Vietnam. It's quite short distance compared to white Thailand or farm Thailand. So on the estimate number here are significant. Yeah. So on the left, you see the 3D figure. It's exactly, maybe we can see that the white Thailand with the yellow purple here is stand on the surface, while the other individual that the out of the surface, like this suggest that the white Thailand, they genetically not a population compared to the other, when the other population, they may be admixture together. Yeah. And on the right, that the population structure, the K number here, I put from 2 to 4. Actually, I put from 1 to 8, but this result is highly have the score high. It means it's similarity score around over 0.9. So we see that if we suppose that they have two populations and the value here is nearly 1. So this suggests that only two populations in this data. And also, if we suppose that the three populations, the answer here, we see three graphs. The white Thailand is stand out maybe very clearly compared to other population, while the farm Vietnam, white Vietnam, they have, how to say, the color yep, admit to the other population. They have the other population. They have the genetic distance yet come from Vietnam and white Cambodia. So we can conclude that in this chart that the white Thailand is one population, distinct population, and other maybe admixture like farm Bangladesh here and farm Thailand, they have admixture to another population. So in this graph, we estimate the migration rate between population that show the genetic share between the population in the asymmetric movement. In the first graph we saw, we used the zero bootstrap method that we just use the contemporary data. And in the second graph, we use the highly resembling data with 1,000 bootstrap. And we see a little bit different between two graphs. In the first graph, we see the high genesis movement between Vietnam and farm white Vietnam. It indicated here, we see that the white and the farm, they nearly much the same in the genetic inheritance. And also the white Cambodia, they have a large genetic share between Vietnam population, while the other population like white Thailand, they has, and farm Thailand, farm Korea, the lower genetic share between the other population, like around 10 to 15%. However, under the significant analysis, we saw that the farm in Bangladesh, they have the genetic come from Vietnam, while farm Vietnam. And also the farm from Thailand, they also receive the genetic share around 8 to 9% from Vietnam, and also the white Thailand, but it's small proportion. But this is very important that we know that the catfish that imported to Bangladesh now, maybe they have the genetic from the local Mekong River. So for estimated effective population size, we try to understand that the long term or the short term of the fitness of the population is like historic effective population size here. And we saw that the value is quite high, around over 1,000 of its population. So just noted that the population with the lowest sample size that I didn't use for the analysis here. So for a long time, maybe around 100 years ago, yeah, the population, they have very high effective population size. And for contemporary estimate, we saw that it dropped too much, like from Bangladesh, they're around 10, and from white Cambodia, around 15. But we found interestingly here, Vietnam stock, they have to maybe higher effective population size, over 50, based on the recommendation for, how to say that, the selective breeding program for breeding purpose, that over 15 is okay for retain the breeding for long. So after the result that we go into three more important findings here. First, that we know that at least two population, but in lower part of Mekong River, and another population like in Bangladesh, like farm Thailand, they have admixture between the other population. And the second is the evidence that of the recent decline in effective population size, like 1 to 1,000 to just 10 or 15. And we know that the white Thailand population, they show the distinct genetic characteristic, and this population may be good for you to form any base population for any genetic breeding program in the future. Adding to that, this study, they have some, how to say that, bottleneck, I think so, how to say, yes, bottleneck, because that the individual between the location collected is not the same. Some they have low number of individuals collected, and some they have bigger, because that the in the upper Mekong River that hardly is not difficult, not easy to collect the animal enough for analysis, and due to the funding, so we just do limited number of individuals. So maybe in much individual is better for the confirmation in the future. I think so. Thank you for your listening. Yeah. Any question?
Thank you very, very much. Unfortunately, we're going to have to move on because you went a little